Hello friends, my dear students, myself Wandhare Sheetal from Rayat Shikshan Sanstha's Rao Bahadur Narayan Rao Borake College Sirampur. This video is prepared under the guidance and support of Honorable Principal Dr. Gaikwad Sahib. This video is only for the educational purpose, as we know that what are the structure of the prokaryotic and eukaryotic cell. In the 11th standard, that all the cell organelles they have given in the detailed form, and these cell organelles in the previous video we have seen that the prokaryotic cell, that is the simple cell, is there, shows the presence of the primitive genetic material, and this eukaryotic, it is the evolved cell due to the presence of the evolved genetic material is present. Now the structure of the mitochondria, chloroplast, as we know that the detail we have seen about that, and what are the roles, function of these cell organelle. The double membranous cell organelle, it is the characteristic feature of this eukaryotic cell, and which is the well developed or the master controlling unit of that cell, that is called as the nucleus. And in this here lecture we are going to show the structure of the nucleus, which are the parts of the nucleus is present, where is it is get located. As we know that here generally the position of the nucleus in the cell, it is present at the center. Depending upon the presence and absence of these nucleus, here the various types of the cells they are present. Some cells they are uninucleated, means contains the only single nucleus. Some cells are binucleated, contains the two nucleus. Some are tetranucleated, contains respectively 3 and 4 nucleus. Some cells are the polynucleated cells are there, they contains the more nucleus. That example of this polynuclear is nothing but here the structure of the neutrophil is there. Here this ultramicroscopic structure of the nucleus as seen in this diagram here. Here that it is the double membranous cell organelle, shows the outer as well as the inner nuclear membrane. Here this outer membrane and inner membrane, it is get separated from each other, and the space is present in between these two membranes, that is known as a perinuclear space. And here this perinuclear space is not the empty, it is get filled with the fluid is there. Inside that here this shows the continuation is not there, the pore is present, means that nuclear membrane there shows the opening, and here this nuclear opening that called it as the nuclear pore. For what purpose? Here this nuclear pore is present for the showing the cytoplasmic streaming movement is get possible, or whatever the substances is present inside that substances, maybe if they have to move outside in the cell, the cytoplasmic bridges they have to get established, and until and up to that equilibrium is get established, here the movement of the substances are there. Okay. Now here the inner side of these nuclear membrane, just like the cytoplasm. The cytoplasm, that is the ground substance of the cell which contains the various cell organelles. Here the nucleus is present, and inside the nucleus that ground substances is known as a nucleoplasm. Here this substance they are showing the presence of the nucleoplasm. Nucleoplasm contains the genetic material. That genetic material, it is present in the form of the chromatin network. Along with that, whatever content is present here, it shows the presence of the nucleolus, it shows the presence of the karyosome is there. Okay. Now these are here the elements or components present in the nucleus is there. Here this nucleus, it is made up, or that nucleoplasm, or also known as a karyoplasm, it contains the various substances. Now which are the substances are present in that? It is shows the presence of the nucleic acid along with the protein molecules, the minerals and the salts. This chromatin network, it is also contains here the U and heterochromatin, how these regions they are get distinguished. Depending upon the staining property here, that U chromatin and heterochromatin they are get distinguished there. This nucleolus, now what is that nucleolus is there? This nucleolus, it is again contains the genetic material. That nucleolus, it is made up of rRNA and ribosomal protein. It is the best known as a site of ribosome biogenesis. Depending upon the synthetic activity of a cell, there are more nuclei may be present in this nucleoplasm. This nucleoplasm, it produces the rRNA and ribosomal protein, which are after that transferred into the cytoplasm and are get together, they forms the ribosome. Now what are these chromatins are there? These chromatin, it is nothing but here responsible to carry out the hereditary, means they are responsible to transfer the genetic information from generation to the generation, from parents to the offspring. This nucleus, it contains here the DNA molecules. As we know that here it is the structure of the eukaryotic cell nucleus, therefore it contains the well identified double-stranded DNA molecules are present in that, and here these DNA molecules they are always associated with the proteins. In a non-dividing cell here, the chromosome they appear as a thread-like network and they cannot be identified separately. They can be distinguished only when the cell it undergoes the cell division. Otherwise, when the cell it comes in the prophase phase, at that time these chromosomes they may look like the condensed form, and just like here that the chromatin network they are looks as the in the condensation form, they are not easily get identified without the stages of the cell division. Now here that chromatin material it contains the DNA as well as it contains the histone and non-histone protein, that is, and the RNA. In some region of the chromatin here DNA it is a more and genetically active. Here in this diagram the actively repeated DNA that is known as a U chromatin, and where the less repetitive DNA it is known as a heterochromatin. This region it can be identified after the staining property under the microscope, which region it is get darkly stained region, that is known as a euchromatin region, and which region it is get lightly stained region, it is known as a heterochromatin region. Means here in euchromatin the more repeated DNA is there, and in heterochromatin here the less number of the repeated DNA is present here. Now here that cell it is get prepared when the dividation is takes place, as the nucleus it is the controlling center of the cell, and itself it gives the order to the cell when it has to go or follows the cell cycle stages. The chromosome they are get in the coiled form, in the condensation form, and during the metaphase here they become and can be identified and they comes towards the equatorial position. This nucleus it contains the total genetic information, and this genetic information or these chromosomes, these structure of the genetic material it is responsible for the transmission and the hereditary characters are there. Here the site of the synthesis of DNA, RNA and ribosome is nothing but here the nucleus is that it also plays the important role in the protein synthesis. As we know the structure of the endoplasmic reticulum is that here the one end of the endoplasmic reticulum is always get attached to the outer boundary of this nuclear membrane, and other end it may be get attached to the cell wall, cell membrane, plasma membrane, or maybe here it will be get the free. This nucleus it is also helps in the phylogenetic study of the animal. The chromosome number from species to species it always remains the constant. Here for example in the human being there are the 46 chromosomes are present, out of that the 22 pairs of the autosomes are there and one pair it is of the 6 chromosome is there. Now here the structure of the nucleus is that. Now come towards the cytoskeleton. What is that? Cytoskeleton. Here this cytoskeleton, it is consist of the microtubules, these microfilaments and intermediate filaments are there. These microtubules, they are made up of the protein, that is the tubulin. Microfilaments, they are made up of the actin, and intermediate filament, it is made up of the fibrous protein. This cytoskeleton, what is the role of that cytoskeleton is that this cytoskeleton, it helps to maintain, gives the shape to the cell, as well as the contraction of the cell, it takes place due to because of this cytoskeleton. The mobility of the cell and the cell organelle is possible because of this cytoskeleton. The change in the cell shape and cell division, these process occur due to this cytoskeleton only. This is the composition and the role of the cytoskeleton is there. Now come towards here the structure of the cilia and flagella. Here these two organs, they are, as we know that, always responsible for the locomotion. This diagram here showing the structure of the ultramicroscopic cilia, how it looks like. The outermostly they are showing the plasma membrane, on inner side the sheath, dynein arms, these microtubules are there, the central microtubules and the radial spokes. These elements, these parts is showing in the structure of that cilia. Now here these cilia is nothing but the very fine hair-like structure, and they are bound protoplasmic outgrowth that are free surface of the cell. They generate a current in the fluid membrane and passage of the material at the time of the locomotion. These cilia, they are having the very minor small insides, and they are having the more number, maximum number of the cilia they are present. They acts as the os coursing movement of the cell, means in short they are responsible for the locomotion. Thus flagella are the longer as compared to the cilia, their number is less as compared to the cilia. They are mostly present in the prokaryotic bacterial cell. The structurally here these are different from the eukaryotic flagella, that cilium and flagellum both on the structure they are consist of here the basal body, basal plate and the shaft. This basal body, it is placed on the outer part of the cytoplasm. It is derived from the centriole. It has a 9 peripheral triplets of the fibril. This shaft is exposed part of the cilia or flagella. It consists of the two part, that is the sheath, axoneme. That sheath is nothing but here the outermost covering of the cilia as well as the flagella. That core, it is known as a axoneme, and they possess the 11 fibrils, that is the 9+2 arrangement of this fibril is there. The central tubule, it is enclosed by the central sheath. This sheath is connected to another tubule by the peripheral doubtlets or the radial spokes. Due to these radial spokes here, these all the filaments, they are get interconnected to each other, and they forms here the linkage or the bridge. Now come towards here the centriole as well as the centrosome. This centrosome, it is usually found in the nucleus, near the nucleus of an animal cell, and it contains a pair of a cylindrical structure that is called as a centriole. When we see here the diagram of the animal cell, in this cytoplasm here we have seen the structure of these centriole. They are perpendicular to each other and surrounded by the amorphous substance called the pericentriolar material. Each cylinder of these centriole, it is made up of 9 sets of the triplets microtubule. They are made up of here the tubulin. Evenly spaced triplet, they are connected to each other by means of here the non-tubulin proteins. All the proximal end of the centriole, here there is a set of tubule. The peripheral triplet, they are connected by this thumb region, and due to this proximal end of centriole, they look like the cartwheel structure. This centriole, it helps in assembly of the spindle apparatus at the time of this cell division. Here we have seen here the role structure of the cilia, flagella, cytoskeleton and the centriole. Now in this video we have shown about the structure of the nucleus along with these other components. Okay, thank you.